Hello, my name is Jacqueline Payton, and I'm an associate professor in the Department of Pathology and Immunology at Washington University School of Medicine in St. Louis, Missouri, USA. I would like to thank the organizers for the opportunity to share my work with you today. The title of my presentation is Finding Balance in the Immune System, Mechanisms that Regulate Infection and Cancer. I have nothing to disclose. As a brief background, I'm a physician scientist and a pathologist. I currently spend my time 75% on research and 25% on clinical service. And my research is translational. We begin with patient samples on which we perform genomic studies to make discoveries that we then follow up with cellular experiments, mouse models, which we then translate back to clinical trials in patients. There are 3 areas of research in my lab. Today, I will tell you about one of them, which concerns the mechanisms of therapy resistance and progression in lymphoma. First, I would like to thank my colleagues and those in my lab who did this work, and without whom none of this would be possible. First, I would like to start with Dr. Neha Mehta Shah and Dr. Amy Musick, who are the oncologist and dermatologist, respectively, who see patients in the WashU School of Medicine CTCL Clinical Center. That is a large nationally recognized lymphoma treatment center that has a high enrollment to clinical trials, and important for these studies, has a long history of banking serial samples from patients pre and post treatment. From those samples, my lab, which is pictured below, performs discovery-based genome, epigenome, and transcriptome profiling studies with follow-on experimental studies in cell and animal models. What is cutaneous T-cell lymphoma? It is an umbrella of several cutaneous lymphomas, which are cancers of lymphocytes that affect the skin. Mycosis fungoides and Sézary syndrome are the 2 most common, and those are the ones that we focus on in the studies that I'm going to tell you about today. As mentioned, these diseases affect the skin, lymph nodes, peripheral blood, and sometimes viscera. Patients with greater than stage 2B disease have only a 20% 5-year survival. There are many therapies at this point, but few are effective long-term. And importantly, there are a lack of biomarkers for therapeutic decision-making. Therefore, the overarching goal of our studies have been to define the molecular mechanisms of progression and therapy resistance so that new therapeutic strategies can be designed and tested. Several years ago, we began with the observation that histone deacetylase inhibitor therapy, although FDA approved, is effective in only 30% of CTCL patients. So we thought, what changes are there in the acetylation landscape that associated with response or resistant to HDAC inhibitors? And if we define them, could we then predict which patients would be sensitive to this therapy? Our first study, led by Jared Andrews, who was a graduate student in my lab, concerned 20 CTCL patients who were beginning a clinical trial for HDAC inhibitor therapy. We collected samples of blood and skin biopsies before treatment started and then subsequently thereafter. We enriched for malignant T cells and then performed genomic, transcriptomic, and epigenomic studies of those samples. This is published work, and so I will just briefly go over our findings. We found a number of pathways that were upregulated at the epigenome and transcriptome level in cell adhesion and migration pathways, anti-apoptosis and growth, T cell activation and immune response. And in particular, I wanted to point out the cell cycle and mitosis and Rho GTPase activation pathways. Subsequent to that paper, we asked ourselves, CTCL patients receive multiple therapies over time, in addition to HDAC inhibitor therapy. And even on all of those therapies, their disease progresses and they show evidence of therapy resistance. So what if there are molecular changes that mark disease progression regardless of therapy type? And can we define recurrent molecular changes so that we could predict in a given patient what therapies they might be actually sensitive to? And so began our more recent study of 35 CTCL patients. We collected more than 100 samples at more than 2 time points for each patient. We performed exome and whole genome sequencing, epigenomic studies, as well as functional studies, RNA-seq and single-cell RNA-seq. These studies were led in part by Jared Andrews, and then subsequently after his graduation by Hannah Durando and Om Katavkar. As an overview, I'm showing here the patients that we call our multi-omics cohort, a subset of 10 patients that had all of the genomic studies performed and all on multiple samples from multiple tissue types, as you can see. Here I'm showing an overview of the pipeline that we used to analyze the exome and whole genome sequencing that we performed on all patient samples, including tumor and matched normal non-malignant cells. We used a fairly standard pipeline for alignment and variant calling using GATK and MUTEC2. We used CNV-KIT to identify copy number variants, and then we annotated these variants using VEEP, and then prediction tools such as SIFT and polyphen. Eventually, we used R and ggplot for the visualizations. This study was unusual compared to most in that it was defined by serial samples collected from each patient, because our goal was to identify variants that were acquired with progression or therapy resistance. Thus, we began by comparing each tumor sample to the normal match sample from that patient to identify somatic variants as opposed to germline SNPs. Next, we compared each of the subsequent tumor samples to the initial tumor sample to identify what variants were gained with progression or therapy resistance at each of these subsequent time points. We found a large number of copy number variations in CTCL, which was known. However, what wasn't known was the number of the copy number variations that were associated with progression, rather than being detected when the disease was first diagnosed. Here in this copy number frequency heat map, the chromosomes are listed across the x-axis, and the percentage of patients with deletions or amplifications in our study group are shown in the y-axis. The copy number variants that were detected at diagnosis in the initial sample are in light pink or light blue, and those in dark red or dark blue were those that were found with progression and therapy resistance. I would like to highlight particularly EZH2 here on chromosome 7, EZH1 on chromosome 17, and STAT3, which were all copy number amplifications that we identified in patients with progression. You can see in 50 to 60 or 70% of patients. In contrast, TP53 was an early loss in about 50% of patients and was detected at diagnosis or the initial sample. So next we looked at the RNA expression level of each of these genes. And here I'm showing the 3 examples that I mentioned on the previous slide. We divided these graphs for gene expression into copy number status. So amplification or deletion, neutral, meaning a normal copy number, and then those patients where we don't have whole genome data on them, and then the healthy donors. For EZH2, you can see that there's increased expression in the patients with amplification as compared to both the neutral and the patients with no whole genome data, and compared to the healthy donors. In contrast, STAT3 patients with amplification or without amplification have increased expression compared to the healthy donors, suggesting that for STAT3, there are other mechanisms that are increasing STAT3 expression, not just the genome amplification. Finally, for TP53, there is decreased expression in patients who have deletion, but also there's some decreased expression in patients who don't have genome deletion. So perhaps there are other mechanisms at play for TP53 as well. Here I'm showing you a large heat map that shows a subset of the mutations that we identified. We selected these by identifying genes that had genome copy number variants and single nucleotide variants, and were detected in at least 2 patients in our group. We then looked for pathways that were enriched in these genes that were mutated, and found an enrichment of epigenetic modifiers, particularly DNA and histone methylation modifiers, such as EZH2, which I pointed out earlier, and EZH1. Also transcriptional regulators, DNA damage and repair, and immune signaling. As I pointed out before, STAT3 is in this group with mutations and copy number variants. We next sought to look at clonal evolution, meaning copy number variation and single nucleotide variation that changes over time as the cancer progresses, using single-cell RNA-seq, because it gives us the ability to identify what variants are present in a single cell and whether the number of those cells with those variants increases or decreases over time. It's a very powerful technique. Here I'm showing an overview of the pipeline that we use for the single-cell RNA-seq analysis. It is, once again, a fairly standard pipeline, at least initially. We use Seurat for QC and integration and normalization. Then we used Copycat to determine a copy number inference from the expression data from the single-cell RNA-seq. We use single R and CB sniffer to map single nucleotide variants to single cells, and then SC repertoire to identify T cell receptor repertoire, which tells us about T cell clonality and is an indication of malignant T cells. We, as before, we use ggplot and Seurat for our visualizations. Ultimately, we had 16 serial samples from 6 CTCL patients. We performed CD4 enrichment to ensure that we would detect single nucleotide or copy number variants in the malignant T cells. And we had a total of over 92,000 cells from more than 2 time points per patient. We initially used Seurat for cell type annotations to define immune cell subsets, and defined various different T cell subsets, including CD4 naive, effector memory central, and some CD8 of the same subtypes. We also identified monocytes and in a small number of B cells. Our enrichment for CD4 was it enriched for CD4 positive cells, but it was not an exclusive sorting. So we did get some monocytes as well, which actually provided a nice counterpoint for the T cell expression. We were reassured that we did, in fact, enrich for cutaneous T cell lymphoma malignant T cells. First, we demonstrate that we had high expression of T cell genes and CD4 T cell genes. So shown by CD4, CD3D, and T cell receptor beta. And then several genes that are shown here, which are canonical CTCL, highly expressed, including TOX, KIR3, DL2, and PLS3. Finally, we used T cell receptor clonality using SC repertoire to identify clonal T cells. So if you're not familiar with TCR clonality, each T cell has rearranged the DNA that forms its T cell receptor, and then it is unique from all other T cells. It and its progeny are unique from all other T cells and actually all other cells in that person's body. And so we can use that to identify clonal populations. In the case of lymphoma, in individual lymphoma, all of the lymphoma cells share that same T cell receptor sequence because they all came from a single progenitor malignant cell. So as you can see, in most of the patients, we did in the pink, I'll direct you to the pink, which are the clonally hyperexpanded. We did successfully enrich for a clonal population of T cells. The one exception is this patient 1137, who unfortunately in the blood samples that we had did not have any circulating malignant cells, which we confirmed with the clinical testing that was performed at the same time and was reassuring actually that our T cell receptor analysis was performing appropriately. We next analyzed differentially expressed genes in the different clusters that we identified by single-cell RNA-seq and identified that in clusters 0 through 8, we again had canonical CTCL genes as exemplified once again by KIR3, DL2, FOXP1, PLS3. There's a loss of CD7, which is actually used clinically to identify and diagnose CTCL. STAT1, et cetera. And then in the other clusters of monocytes, we see monocyte canonical genes such as CD14 and CSF1R. To demonstrate the clonal evolution that we identified in these patients, I will show two examples of copy number alterations and single nucleotide variants that we identified in two different patients. One of them, patient 1084, had fairly stable disease and was responsive to therapy. So the pink triangles indicate therapy change. And you can see there's a new therapy here, a change in therapy here, and that's all. And then eventually this patient died from unrelated causes. In contrast, patient 1126 had many therapy changes and died with due to progressive cutaneous T-cell lymphoma. To demonstrate that we did successfully enrich for malignant T cells in each of the samples, 3 samples that I'm showing you here, where we did single-cell RNA-seq, the malignant T cells are labeled in pink and the non-malignant T cells are labeled in blue and non-T cells are in green for each patient and each of the samples. The percentages of cells are quantified here, so you can see that indeed we did enrich for malignant T cells as identified by T cell clonality. Here we're showing copy number alterations that were present in these patients and how they changed over time. In each case for these genes, patient number 1084 did not have a copy number alteration. Their disease was copy neutral. And so the cells are all colored in kind of this yellow, which is a copy neutral, neither loss nor gain of these loci. In contrast, patient 1126 has a gain of EZH2 at this earlier time point, which is here. And then at later time points, there are fewer cells with gain of EZH2. And this is the second time point here. I'm sorry, that was the first whole genome time point. The first single cell time point is here, and then the second single cell time point is here, and the third is here. And so you can see overall, there's actually a loss of these cells that had a gain of EZH2. And that coincided with differential therapies that the patient was receiving. In contrast, there is a deletion of CDKN2A, which is associated with cell growth and genome replication. And that one is fairly stable over time. All of the copy number changes are quantitated over here in these violin plots. And purple is the 1126 patient, and teal is the 1084 patient. And the last example is for PDL1, which is a target of checkpoint inhibitor therapy, which you may have heard of. It's a type of immunotherapy that has been more recently developed. And so this patient 1126 received it later in the time course of their therapy. And you can see that, in fact, over time, there's an increase in the number of cells who have lost PDL1. So an indication that these cells have lost the target of the immunotherapy that this patient was receiving. And that may have been the cause of the therapy resistance that this patient experienced. Here we're showing single nucleotide variants that were detected over time. These are the same patients and the same single cell data sets. 1084 had 3 single nucleotide variants that we're showing here. There was some variability in the amount of the DUSP5 variant, but overall, they're fairly stable numbers of cells that have expression of the variants that were detected, which is consistent with the fairly stable disease that this patient experienced. In contrast, patient 1126 had increasing numbers of cells with different variants that were detected, including BCL2A1 associated with cell growth, and PIK3R1, which is associated with PI3 kinase signaling. In summary, I wanted to just tell you a little bit about our ongoing work and future directions. The identification of EZH1 and 2 was really striking to us, and importantly, there have been a recent development of EZH1 and 2 inhibitors that have shown efficacy and minimal cumulative toxicity, which then means for the patients a fairly favorable quality of life, because with cutaneous T-cell lymphoma, unlike with other cancers, patients actually have to take their therapy for long periods of time. And so it really needs to be a minimally toxic therapy for these patients. So about more than a year and a half ago, we started with Dr. Mehta Shah leading this, started a new investigator-initiated phase 1 study of tumaminostat, which is an EZH1/2 inhibitor in patients with CTCL at WashU. That study is currently ongoing. And then for my part in my lab, we are continuing with mechanistic and animal studies of mutations that we identified in this multi-omic project, including EZH2, TP53, and STAT3. I would like to thank, once again, the people who work in my lab, Jared Andrews and Hannah Durando, and Neha Mehta Shah and Amy Music, without whom these accomplishments would not have been possible. Of course, I would also like to thank our funders in the Siteman Cancer Center, Leukemia and Lymphoma Society, the Foundation for Barnes-Jewish Hospital, and the NCI for their support. And of course, without them, this also would not be possible. And I want to thank you for your attention today. And I've been very grateful to have the opportunity. Thank you.